the founder of Bioclues, which invariably has been existing over the last 18 years. And we have been organizing, this workshop over the last, I would say, uh, uh, 3-4 years in the name of integrated systems bioinformatics. But over the years, we have organized several workshops and several events. But the main goal of this particular workshop is to bridge the gap between biologists and information technologists, largely at the coalesce of boundaries that we try to bridge the gap between PhD fellows who want to take up good science. So what we have been doing over the years is we invite several faculty members across India and the world. We give a gist of certain topics which we are going to cover in the next 2 weeks. We give a kind of an orientation in the form of a theory and some kind of hands-on spanning for about 45 minutes to 1 hour. And then after the workshop is done, we encourage the participants to do a kind of a mini project. Okay, and this particular mini project, will be taken up, with all the faculty members who are going to teach during the next, 4 months. So which means this workshop is indirectly associated for over a semester, I would say. So let's assume we finish this start program from July 17th till 29th. And probably from August 1st, we will have this, what do you call, projects that we are going to start with all the, faculty members based on your choice. There will be enough room, for you all to discuss the problems that you're facing as a part of your project. So that, each one of us can help you overcome certain problems that you're facing as a part of your project goals. And this is only for life members, so which means you pay a small amount of 2,000 rupees to become a life member of Bioclues, and then you attend this. Okay, and there will be enough room for all of you to guzzle some kind of freedom in asking questions. This talk, all the talks are recorded and they're put up in the YouTube page of Bioclues. Thanks to Goldie Singh, our student chapter chair, who is recording the sessions, all the sessions. And the program looks like this. So today, July 17th, from 6:30 to 7:45. Excuse me for about 8 to 10 minutes because I have a shipment coming from the UK. I need to store it in minus 20. I'm expecting a courier, to arrive now. It's coming a bit late, so I might be, away, for about 8 to 10 minutes. So I'll be discussing about bioinformatics and systems strategy. And today, please install Cytoscape with CytoHubba and ensure Java is updated on your computers so that I'll give you a quick demo. Jeraman Valedi, who is our advisor-in-chief, sir, needs no mention. He'll be taking you to the methods in machine learning. So please download LibSVM software. Sundararajan Sundarta, who is based in Singapore, he is going to give a gist about data science. And for that, you download the Veka software. And again, on July 20th, I'll be taking the essentials of NGS. So please create an account and use galaxy.org. Usually, we have several pipelines in command line, which I usually teach as a part of a PhD course, but since it's going to be a small gist, please create an account and use galaxy.org. And we have Rohit here, who has been a long-time associate, and he is a VIT alumni, and was an ISCD student chapter chair, International Society for Computational Biology chair. He was also associated with Bioclues and currently is in the States. He is going to give us a gist of analysis of epigenetics and chromatin data. Okay, and all these things, I think we have created a sheet here which is open to you all, which we have shared with you here. Okay, so this particular thing I'll probably share it here in the group as well. I mean the WhatsApp chat as well. We're going to create a WhatsApp group as well. So I'll get back to you on this. Okay. Uh, and then we have Sunday. Of course, Sunday is off. So have a good Sunday. And then we come back on Monday with Sandeep's talk on assistance after infectious diseases. It's largely towards the microbiology perspective because we need to cover the microbiology aspects as well. And subsequently, Renuka is going to take on molecular docking and simulations, where she will probably take up some kind of SARS-CoV-2 and ligand receptor based docking. Excuse me, I just need to pick up my this thing. Indrani, please, take over, please, for a few minutes.
Hello, everybody. Goldie, is my voice audible? So, as the sir has mentioned, so you have seen the schedule, right? So, we'll be having a detailed overview, not only overview, but in-depth concepts. What are the basics, right from the basics to the advanced uh, courses of this system biology workshop? You'll have a great idea. And as Prashanth mentioned, that you'll be given with a workshop in a practical session also. So we'll ensure that you are being given a maximum exposure on each of these topics that sir has mentioned. And with the project topic also, we will enable you to, in some cases, we will try to publish those. Okay, so with your cooperation and all, we hope we will make it a success and you all enjoy this learning process. Okay, so wherever you have any doubts, anything, you will feel free to contact each of the mentors that sir has mentioned. So at any time, they'll be there to assist you in any ways. Okay, so far, any doubts? Okay, so can I know like what all backgrounds you have, UG, PG, and what are beyond that you are all pursuing? Rakesh, yeah. So can you please introduce yourself, please?
Yeah, my name is Rakesh, and I have a BSc in Computer Science and Bioinformatics and MSc in Bioinformatics from Pondicherry University.
Oh, okay. Are you
Cur-
pursuing now or you have finished MSc?
I have finished. I'm doing right now a job at Mr. Biologist LLP in Guwahati, Assam, as a bioinformatics analyst.
Okay. I'm also an alumni of Pondicherry University, but I did my graduation in and post graduation in molecular biology and biochemistry from Pondicherry University. Okay, and I think I know Pragati. Pragati, are you there? Okay, who else have gotten exposure to? You can just raise your hands and tell who have gotten exposure to bioinformatics and advances, even basics or advances of bioinformatics. You can just raise your hand. Okay, it's quite pretty number of people who got an exposure into it. So it's good. It's good. It's pretty good that you all have been on. Okay, okay, fine, fine. Okay, it's pretty good that most of, I mean, 50% of people have gotten exposure to bioinformatics, maybe basics or maybe advanced. Uh, but this course will make sure you will be understanding the concepts of machine learning. Mostly, our chief guest, like as Sir mentioned, Jaraman Sir. He's an expertise in that field, in and across concepts of machine learning and artificial intelligence. His main area of interest is based on that. So uh, you will learn a lot. I mean, I can make sure of that. And if you face any contacts, not only after this workshop, also not only during this workshop, you will stay in contact with each of the mentors. Uh, I should introduce myself. I'm Indrani, and I am actually pursuing my PhD final year. I'm an ICMR research fellow at Pondicherry. So, uh, as part of this workshop, our secretary, like Bioclues secretary Manjari, Dr. Manjari, and myself will be taking care of that part. So that is during the end of the almost it is coming around July 26th. Yeah. So the next week. So each of these courses, whoever have not gotten exposure to machine learning and all, you will get a very you know how to say key insights you will be understanding from him. But before that, if you have not read about artificial intelligence or have not undergone, so you can just have a brief look onto it. Okay. Mostly it is dealt with the programming languages R and Python. These two programming languages are like key roots of handling artificial intelligence or machine learning models. Okay. And fully we see you like you don't get bored or you enjoy this process. Who else is there? I see people with chromosome. One of the bands being highlighted. Who is this? Can you just introduce yourself, please?
Hello. Yes, ma'am.
Yes, sir.
I'm audible.
Yes, yes.
Yes, myself, Dr. Vipul. I'm working as an assistant professor forest biotechnology. Uh, ac- and I'm working on plant architecture.
Oh, okay, great, sir. Great to hear
Yes,
from
and
you.
recently
Okay.
we got project under which we are developing a omics facility. Earlier, I worked majority in wet lab. So I would like to learn more about this bioinformatics and related things. So this is my.
Sure, sure, sure. We hope that this entire workshop, I mean, whatever topics we have taken, uh, each of these concepts will make you understand. I mean, at each part of bioinformatics, you will be able to apply in your subjects also. We have many student chapters here, Dheeraj, he is an alumni from Vignan University. Recently, last year, Index was conducted in Vignan University, sir.
Okay.
So it was, I mean, yes, hoping to, if you can look into the, we'll soon publish all the brochures and all the details. So you can have a look at Bioclues website also. So
Sure.
you can see for the past events that we conducted,
Yes. Sure.
including we have our major outreach and entrepreneurship chairs also,
Definitely.
who will help you like in assisting as possible. Mm, like you can go through our websites. Apart from that, we publish every quarterly newsletters. What all activities that we conduct every 3 months that gets we publish in the Bioclues website, and we have all the scientific conversations also. So all of the participants are being encouraged to attend and see each of these cyber sessions. If you can attend, uh, it will give you a very you will always know that learning never ends. Okay, so it is basically we all are from different backgrounds, but the science where it is leading to, we always have to focus on interdisciplinary sciences. It is not focused in one single discipline. Okay, so that is what I want to say. All the best for all of you. And Sir, Professor will take over the classes for, I think, the NGS basically. So, Sir is an expertise in system biology and our full and full he is on to it. I know I hope Rohit sir is very much acquainted with him. So he's an expertise in that field. I have already mentioned about Jayaraman sir. Then we have Sundarajan sir. He's also an expertise with a strong background in bioinformatics, and he will be teaching you about the data science and its importance with relation to bioinformatics softwares. And Manjari, yeah, I forgot to mention about her, Dr. K. Sri Manjari. She is actually secretary of presently for Bioclues. She held her PhD in genetics from Osmani University, and she's presently as working as a scientific editor in Gene Powerx Hyderabad. And she's very dynamic. I must say. So Bioclues, I mean, it has the Women in Biology platform. Those who are interested to become the light members, you can certainly. I think you have all registered and came, if I'm not wrong. Is that so? Can I get a yes or no response from the participant? Hello, hello.
Yes.
Yes, ma'am.
Ma'am registered.
Oh, okay, okay, sir. Okay,
Yes.
sir. Thank you, thank you, thank you so much. So you will get to attend hereafter. Once you register this light memberships, that is why we gave the term as light members. So once you get registered, you will be you will be allowed to attend all the other workshops free of cost, and all these cyber sessions that we have monthly. We also do have this other. We bring as actually we bring in a lot of activities that we have student chapter especially. We have now so many student chapters, including one international student chapter. So we try to bring more and more students. This is all in and across faculty, students, whoever are lying in wherever backgrounds. We try to inculcate the knowledge in such a manner that people don't stay lack of the research that is progressing. Right. And nowadays, if you look in publications, it is quite difficult because we are not able to reach on that mark. So I hope you all will enjoy this process. Just give me a minute. So we'll start with the session. I hope you have all entertained about the whatever I mentioned about Bioclues, and if you are still more curious to know, you should not fall back from visiting our website for the latest updates. Prash sir, are you there? Prash sir, am I audible? Prash sir? Thank you, sir. I think there is some network issues from sir's side. Uh, just give us a few minutes, one or two. Maybe we'll get back to you with the session. Sir, are you there?
Yeah, I'm sorry, I'm just back. Yeah, 1 minute.
Yes, sir. No problem, sir. I have just mentioned about our activities that we pertain in Bioclues and all the possible uh, chapters that we have, everything in like in details for our Bioclues and about the workshop also, sir. Are you going to take over the session, sir?
Yeah. Thank you.
Yeah. Oh, thank you, sir.
Yeah, first of all, I apologize, dear friends. I'm really sorry. I had a major shipment coming from the UK. My office is in third floor, and I had to come down. The shipment was in Kochi, and it was taking some time. Yeah. I hope my voice is audible, everyone. Yeah. Hello, sorry. Is my voice audible? Yeah.
Yes, sir. It's completely audible.
Yeah.
Yes, yes.
Thank you. Yeah. So where were we? Yeah, I think we were here. Yeah, and Manjari will take meta-analysis. Oh, I didn't share the screen. I'm sorry. Manjari will take meta-analysis review part 1, followed by Indrani on 28, part 2. Meta-analysis is largely towards how to engage data from different sources. And then Anil will take up plant bioinformatics. And finally, we'll have a summary and quick projects and feedback. Okay. And please don't edit anything here. We will share the links to you separately. And we will have an attendance here. The attendance for all the 2 weeks is a must in order for you to get the certificate. And kindly, honor the time of the faculty here. Okay. Yeah. So we will create a WhatsApp group because the reason why we didn't create the WhatsApp group is we wanted to see how many would really turn up because we had about 68 registrations and I could see there are about 20 participants here. That is expected because we have about 400 live members and about 68 participants registered for this and could see only 20 attending. So moving further, so the learning objective for this particular thing is for my part of the course is that we will learn the interactions within the biological networks and we quickly learn about the next generation sequencing based approaches, how systems biology can really be a prelude, for next generation sequencing. That is what we'll probably learn during the next, 1.5 hours. And this will also subside with my talk on 20th as well, where I'll be talking about the next generation sequencing. So what I'm doing is I'm covering these 2 units in 2 different days. So unit 1 is about protein-protein interactions and systems biology and regulatory systems genomics, some kind of hands-on which we'll be covering today. And then, of course, as a part of Unit 2, we'll learn about the next generation sequencing-based approaches, the whole exome sequencing and whole genome sequencing. And of course, that will be taken up by the advances in single-cell genomics and transcriptomics by Rohit Reja, where he'll be covering some aspects using online-based tools. There are certain tools that he's going to give a demonstration. So this comes as a perfect, fitting. Okay. So Rohit, are you there? I mean, I just thought, I wanted to ask, would you also give?
Yeah.
I was wondering, Rohit, would you also give them a demo using Galaxy or how is it?
I think I'll focus more on the downstream tools. So it will be mostly after you have done, the peak calling and then what you can do with those peaks and how you can visualize the data. So most of the downstream analysis is those online tools I'll be focusing on.
Right. Yeah. Just before we thank you, Rohit. Just before we start, we usually have a pre-evaluation done so that we'll know the girth of the participants, how they are good at programming and other things. We have a form which we have created some years ago, but which we keep on updating it. So I'll be giving this right away. So please fill this form during the next, I would say, 3 minutes, which I'm going to share the screen. Give me a minute. Yeah.
Sir, are you mailing it?
Yeah. I'm just loading the link. Give me a link. Give me a minute. Yeah. Yeah, so this is the link. So please fill the form. It just takes 3 minutes for all of you to fill up. Okay. Yeah. So you have 2 more minutes to finish this, please. Huh? Yeah. Yeah, 1 more minute, please. Yeah. Yeah, we are closing the link in about 2 minutes. Yeah. Yeah, about 20 seconds, please. Huh? We'll be just closing the link, please. Huh? Yeah. We received about 15 responses now. Yeah, 16 now. Yeah. So thank you so much. So this is a quick outline of this thing. Many of you don't know about basic NGS. A few would know about math logic and basic Linux programming. Many of you come from wet lab molecular biology, as you could see here. Okay. Many of you know very essentials of bioinformatics. Many of you are very likely to use Galaxy, and most of the things we will be covering using Galaxy as a part of the project as well. And I see many of you do not have any sequence data archive data sets, which we thought we will be using it as a part of our exercise. But nevertheless, you can send us a link later. So this was a typical scientific question we usually ask. So what's the odd one out? Okay. So DNA transcribes, mRNA translates protein. This is well known. This is central dogma of molecular biology. Okay. DNA protein, which means DNA amplifies DNA, which is DNA describes the DNA within, and then you get a particular protein where your transcription is silenced. This is also expected in nature, okay? But never in nature you expect a kind of a DNA that is obtained from a protein, okay? So this is an odd one out. So only 21.4% have answered probably correct, okay? So this is the correct answer. And some of you have given this feedback, which will probably keep you posted, okay? So so what we'll be doing the next, I would say, 1 or 20 minutes or so is, we'll talk about some aspects of biological networks and some kind of high throughput methods in the realm of systems biology. We'll also learn, some kind of tools, tricks, ticks, and traps with the networks, some kind of networkology concepts, largely taking some exercise using Osprey and Cytoscape, which we will probably do it. Ingenuity, of course, no, we don't have it's a PAR licensed version, so we don't have that. We respect open access, so we'll be probably using some of these other tools. So most of our talks, all the talks are self-exemplary. You have certain fonts at the, you have certain, references at the end of every slide, okay? If they are not, of course, that's author's own result or, figure or image. All the presentation slides will be shared with you at a later point of time in PDF, so don't worry. We'll make it more interactive and make it more personalized as well. So during the part one, what I thought was I'll quickly tell you about systems biology aspects. So if we talk about central dogma of molecular biology, we talk about DNA transcripts, mRNA translates to protein. Likewise, we also talk about central dogma of bioinformatics, where you have all the biomolecules within, be it DNA, RNA, or protein, and you can probably ascertain the function of these particular protein molecules only when they get folded. Okay, so which means you expect a protein to fold, and that particular protein, as a result, invites some kind of ligand to bind to that. Okay, or you have certain groups that are open. These particular yes, sorry, Manav, I'm sorry. Yeah, sure. I'll speak a bit louder. I don't know why. How about now? Is it better? I don't know why. Is my voice better now? I'm sorry. You can use your mic. Yeah.
Yes, sir. It's better. It's good only.
Yeah.
You can carry on. Yes, sir.
Yeah. Yeah. I'll have my energy drink so that I can speak better. Yeah. Thank you. Yeah, so so you have those particular groups or ligands to bind to those particular regions, and that is where your particular protein might invite certain groups or certain pockets where a ligand or a drug molecule can bind to that particular region. So in nature, if you have a sequence, the sequence should really fold, and then only that particular foldable structure can represent a kind of a function. Okay, so that is what we call it as a central dogma of bioinformatics. Nonetheless, not every sequence folds properly, or not every sequence folds impromptu properly. Okay, which means you cannot expect a sequence to fold every time, and as a result, because of that foldability, it invites a kind of a drug molecule to bind to that. Okay, so there might be misfolds, and in nature there might be disorderliness of that particular protein sequences as well, which means the structures might be absolutely in a haywire, where it may not really be in an ordered fashion at all. Okay, so that kind of disordered proteins also do exist, and obviously when you have such kind of structures that are misfolded or disordered, obviously you don't have any function. Okay, so sequence and structure and function is an antithesis to what we call it as a central dogma of bioinformatics. Whereas sequence predicts structure, structure predicts function, and for many of these particular sequences, if they are hold it properly, then obviously you can get the functions of these kind of things. So in nature, you also see what we call it as a Vis-a-vis principle. Vis-a-vis principle is in IT, what you see is what you get. Okay. In fact, in a Vis-a-vis principle, it's just like what you see in the app-based things. You are using a mobile phone, you simply click it, and what you see is what you get, which means you click it, and that is what you get it, right? So in nature, if you have a sequences, if you see a protein sequence, that particular sequence is as a result of certain domains that exist across the length and breadth of that particular protein structure. Okay. So for example, you have a protein like this, you usually have 3 domains, N-terminal, middle terminal, and C-terminal domain. Okay. Usually, each of these particular domains play a very important role because these domains are extra large, by event of which they are extra large, they are folding. Okay. Whereas domains harbor motifs or short signature sequences, because if the domain folds, the motifs or short signature sequences also fold. Okay. That's the beauty. And obviously, if the domain doesn't fold, these particular motifs or short signature sequences do not fold at all. Okay. So this is where the central dogma of bioinformatics would really come into picture. And as always, the foldability or the folding potential of that particular protein depends on several factors, largely the environmental factors. How that particular protein folds properly. Okay, how the protein probably has a capacity not to fold, or in different conditions. Maybe there might be certain proteins that might be overexpressed in certain conditions. So it might be folding rigorously, okay, than compared to the others. So that kind of things do exist in nature as well. And that is largely dependent on the epigenetic spectrum, how the environment will change, how the protein undergoes post-translational modification, which means after translation, there might be a lot of acetylation events, methylation events, okay? All those kind of things play a very important role. And this is literally an old slide, but it doesn't make any big difference. As you could see here, if you see GenBank is NCBI, which is a trademark name. You see the number of bases are increasing almost at a greater pace. As you could see in 2000, you have got about 10 lakh bases, at that point of time, because you don't have many. But now you could see 1 billion bases, in fact 10 billion bases, as we have crossed 2023. The number of sequences have also increased to over 1 to 2 billion. That increases us with the number of whole genome sequences that are being updated from time to time. Okay. But there's something called Moore's law. Gordon Moore, who passed away 2 months ago, he says that there's something called Moore's law. Moore's law says that in computational technology, the computability gets doubled every 2 years, whereas the data that is stored, okay, to compute that, okay, is also getting doubled. But the price to acquire or the cost to acquire that particular space is reduced to half. So in a nutshell, say if you're using a 4 GB computer today, maybe 2 years down the line, you might be using an 8 GB RAM computer. Maybe the cost would be the same, because that's where the Moore's law is attributing to, and that's where in analogy, the number of genome sequences are also increasing, from time to time. That's where this systems biology has taken its birth years ago. In fact, it has taken in the form of a rebirth. It has been existing of good old days, where we have been talking about a system. A system could be a computer system, an organelle system, or an organism, where we as humans, we have got all the body parts that are connected together. We are a human system. Likewise, we as a system has got the major components and minor components. Okay, major components in the form of what we see like hands, eyes, legs, the body parts and other things. And each of our tissues have got cells. There are about 1 trillion cells in our body. And each of those particular cells has got a genetic material in the form of a nucleus at the center. Okay. And all those, kind of things which we have learned during our, good old days. But over the years, thanks to these umpteen number of genome sequences that have been available, there have been mutations that are arising and falling. There have been proteins that are evoluting and devoluting. The interactions interacting, the strains that are probably adapting and fitting. Okay, the best is about the SARS-CoV-2. As you could see, we have got about 17 different SARS-CoV-2 variants, in the form of lineages that have really come across, thanks to the evolutionary landscape that it has heralded. Okay, in that particular way, not necessarily the same mutation exists in one particular variant. Maybe you might have different mutations, keeping on, existing, from time to time. Okay? There are, genes lost and found as well, as a result as well. Okay? So I would say systems biology is lost into oblivion, and that is where, this bioinformatics necessitated systems biology is really taking place. Okay? So, so in nature there are contexts. Contexts are nothing but ohms. Ohms are nothing but many. So whenever you say genome, it's nothing but study of many number of genes. Proteome, study of many number of proteins. Okay, so on and so forth. And every aspect of these is enabled in the form of, omics. So for example, if you talk about plants invite a lot of stress, being a sessile organism, right? Animals, invite a lot of food deficiencies. Bacteria invites a lot of, whether or not them, monoisolates or multi-isolates. They might be pathogenic, non-pathogenic in nature. They might be host-specific and host non-specific sometimes. Okay, but we humans, as an intelligent, species, we are always, surrounded by, several factors. Largely, we know how to combat the stress, how to combat the environmental disturbances, how to combat the epigenetic spectrum, how to combat the epigenetic landscape. Okay, thereby, different phenotypes that we can herald with different diseases. We can, we have learned how to combat that, and that is probably the reason why we generate the data coming from these particular samples. We get a lot of data in the form of true positives, false positives, true negatives, and false negatives. But at the end of the day, what we really need is, whether or not from false positives can we predict how many of those particular false positives are specific to that, so that we can get the true positives. And that is where our lab, systems genomics lab does. We do a lot of, multi-omic integration-based approaches. We collect the data. We take the genomic-based sequences. We take up, clinical data. We make use of it very effectively. We apply it. We try to integrate them and come up with certain, sequence-based approaches. Okay, so this is where we need to look into those things. And likewise, to perform the task, to analyze these particular data sets, there are plethora of tools. Princeton University alone has developed 3 wonderful tools like Seek, Giant, and Imp, okay, which are largely used to integrate such functional networks. We have a Galaxy project at Use Galaxy, which we'll be using by and large for at least 4 or 5 exercises today. During the next few days. And then we have had genome space. Unfortunately, genome space is now closed. We have a C-Bio portal, which is a cancer bio portal, which is a wonderful tool. And of course, the BioGrid, which uses these particular things. So a quick summary here is bioinformatics necessitated data is very important, which plays a very important role as a prelude to systems biology. In order to use bioinformatics necessitated tools, we use integrated systems genomic approaches. And we need collaborations, not really, the competition that we need to herald between bench scientists and probably other biologists. Sometimes we may have to look into the pipelines, not only the tools that we have discussed like Seek, Imp, and C-Bio portal and other things, but sometimes we may have to go beyond and ask certain questions like, are there any kind of integrated tools like spatial temporal interactions? And then sometimes we may have to look into the next generation sequencing based approaches. So keeping in brevity of time, I'll quickly move from system. As I told you, system is nothing but a theoretical entity where we talk as a unit. So we as a human system are compounded by several things. As I told you, we have several entities within each of these trillion cells, like small molecules in the form of DNA, RNA, and protein, where these are integrated and interacting with each other. We have got those strains adapting and unfitting in environment, mutations arising and falling, proteins evoluting and evoluting, the genes lost and found, and interactions interacting with each other. Okay, all this kind of thing is systems biology. And then right from 1859, Charles Darwin's Origin of Species, we have had amazing amount of data that has been produced, but that was not when NCBI was founded in 1988, the National Center for Biotechnology Information. Finally, in 1994, the website was launched where the internet came into existence. And ever since, several genome sequencing projects came into picture. Several evolutionary landscape and mechanisms have really come into picture. Several genomes have been sequenced. Several known unknown regions in the form of hypothetical proteins have been existing. And I've been a part of this bioinformatics.org, which is the world's largest bioinformatics society, and I've been an associate director since 2001. This has been where all our thought leaders convened, and we set up this open source institute. But now we are in a post-genomic era. We are talking about latest next generation sequencing, or the latest third generation sequencing based approaches, okay, using the high throughput methods and all of them. And then we have been talking about the systems. As I told you, the major system includes tissues or organelles or organs, or the minor components includes the metabolites, genes and proteins. I'm sorry, Vijayanta, how about now? Is it better? Yeah, I'm really sorry for this. Yeah. Otherwise, I'll use my other mic. I don't know why I'm getting this problem.
Yeah. 1 minute.
So the second one is very clear, sir.
Yeah. How about now? Is it better?
Yeah, better.
I'm sorry for this. I'm using my mobile microphone. I don't know this. I have a camera. I have a web camera, a wonderful web camera. But I don't know. Sometimes the mic doesn't work. Yeah. So we have these major companies and minor companies that keep on interacting with each other. So it's basically a dry system with a wet biology. We always try to ask how these particular small components or large components they keep on interacting with each other. Nonetheless, when these small molecules like proteins they keep on interacting with each other, they give rise to functions. So, for example, if two proteins are interacting like this, they give rise to some function. Okay, that's where we talk about the survival of the fittest. Okay, right from Charles Darwin's Origin of Species, where we knew that a larger animal might want to take up or eat a smaller animal, another smaller animal, and so on and so forth. But that's right from the origin of species, survival of the fittest. But today, we are talking about arrival of the fittest, which means at what amount of time an organism can arrive in an environment and can sustain. The best example is SARS-CoV-2, as you all know. SARS-CoV-2 is a classical example. Okay, you see, it has been sustaining over the last, I would say, 3.5 years. COVID is still existing in some part of remote parts of India. Okay, where there is no hygiene. So it has really taught us to be hygienic, so that it has given us a kind of a lesson, and it tells us, hey Prash, or hey human being, if you're not hygienic, then I'm going to attack you again. Okay, so that is because it is arrival of the fittest, which means the organism has learned certain mechanisms, okay, to understand the kind of immunity that is pervading within these particular human beings. It has known how to kill that kind of immunity, or how to escape that kind of immunity, so that it can keep on causing those infections. And that is where we can learn about the omics. As I told you, omics is many. This is a classical example which I keep telling my class. So whenever you talk about omics, as you could see here, Um, at the base, you have the genomics. Thanks to the alternative splicing, not the entire genomic reporter gives rise to the same amount of the proteomic reporter, which means if you have a 1x size of a genome, not necessarily the entire open reading frame gives rise to only one protein. Several open reading frames give rise to several proteins. So there might be a 2x compendium of a proteome here. And you have got a metabolome, which is metabolites, where several metabolites keep on interacting with each other. Okay, and there's something called metabonomics. Metabonomics is the data that is coming up from metabolomic data. That is a metabonomic data, and they keep on interacting with each other. And finally, not all interactions give rise to functions. So basically, the take-home message from this theory that I have hypothesized here is: if you have a genome of 1x, invariably you have a 1x size of a functome, which is a functional reporter. Okay, so that is what is the omics limelight. And of course, for this, there are different tools and databases. There are different types of systems biology based approaches, largely in terms of reductionist based approach and dynamic based approach. So dynamicists are simply rely on the systems theory. So it is what is known in theory, but is not probably relevant to practicals. Okay, maybe they might have developed a kind of a model hypothesizing one theoretical entity, and they come up with a conclusion and they say that yes, this model can be probably converted as a network, and then we can employ our survival of the fittest theory or arrival of the fittest theory, or make up some kind of machine learning heuristics, some kind of data that is invariably coming either from quantitative assessment, or sometimes making up some qualitative assessment. Okay. Whereas reductionist approach is purely from a wet lab perspective, largely from a molecular biology perspective. So it's based on the high throughput methods. So which means you perform an experiment, you probably rely on certain data, but the data comes later. But you collect the data, you probably sequence it, you perform certain experiments, and then you ask a question: what is that particular data is going to do? And this is what Hugh Angus in 2003 has developed from protein-protein interactions network. So the loose definition for systems biology is application to projects that are making up the individual biological networks. Whereas a broad-end and derivative network is a multi-integrated approach, taking up theoretical models, statistics, AI, mathematics, engineering, and all those kind of things. So in an integrated systems biology, you talk about the omics, the omic-based approaches, the gene expression patterns, or the metabolomic patterns, or metabonomic. As I told you, metabonomic is a data integrated metabolomic approach, or the phenomic. Phenomic is nothing but the phenotype-based approaches. So all those kind of things can really taken up these kind of things, but not necessarily when you talk about systems biology and ask certain question, what happens to those molecules that are interacting with each other. You also come up with a conclusion and say that yes, what is in theory need not fit practically. Okay, and a hypothesis-driven approach need not always be an experimentally driven approach. If there are interactions known, we can always take measure of unknown interactions. Okay, so that kind of things can really be taken up in systems biology. So, for example, I'll give you a classical example in the next following slides. You zoom in within the cell, and you assume that there are a host of proteins that are interacting with each other. Say A interacts with B interacts with C interacts with D. So in protein interactions also, the transition theory applies. So if A is interacting with B is interacting with C, maybe there is a likely chance that A could also be interacting with C. Okay, so that is where the protein-protein interactions can play a very, very important role. And this is how the definitions are suited. So you talk about molecular biology, you talk about computational biology. Okay, it's very easy definitions, but certainly different. But when you talk about molecular systems biology, you are asking how the molecules work in a system. Okay, whereas in a computational systems biology, you ask how the molecules work in a system using integrated in silico based approaches, and that is what we really define systems biology and computational systems biology. And whenever you talk about protein interactions, there are always traits. Okay, as I told you, for animals, maybe if an animal is fed properly, then only probably it sustains. Okay, like how I'm probably having a sip of tea here, green tea here, and how am I able to talk, because it gives me energy to speak. Likewise, sometimes we may have to ask a question, like if an animal is overgrazed, like what we have asked in terms of our diabetes related question when we were working on animal feed efficiency traits. If an animal or if an eutherian like cow is overgrazed, does it give more production of milk when compared to a cow, or as opposed to a cow that is not probably overfed? Okay, so what are those kind of things that we need to ask? That kind of traits that we need to ask. But here in a protein-protein interactions, there are traits per se largely taking up into the organization, how these proteins keep on interacting with each other. Okay, how they sustain the networks, how they sustain the interactions, and that is where the protein-protein interaction network came up in 2014, and after that, not many papers have really come up in proteomics, because large focus has really steadfastly moved towards the genomics, because next generation sequencing came up in 2009, and that is how a large scale protein interaction based networks have really come. So this is what, say for example, you have a protein here at the center. Okay, I hope all of you can see the screen, right? Yeah. So you have you have a node here, which we call it as a protein here, with SP. It is known to interact with several proteins, and this is represented by different lines in the form of edges. These are called as edges. Okay, and each edge color represents different ways to represent a kind of a network. Whereas here, as you could see here, this TPIA is known to interact with some hypothetical protein like PGK or something. And it is known to be interacting with several lines, which means there have been experiments deciphered to show that these two particular proteins are known to interact by different experiments, maybe one using histohybrid screen, one using a simplest pull-down assay, using co-immunoprecipitation, or using mass spectrometry. There might be different experiments. Sometimes using neighborhood as well. Maybe a gene might be present adjacent to each other. Then you may say that if they're adjacent to each other, then they might be interacting as well. So what has really been existing over the years is, if this is my network, can I probably understand the known unknown regions in the genome? Okay, that is what my lab does. So that is what our lab has really been shown to interest. Okay, so as you could see here, you have a protein here. Protein is known to be interacting with another protein. Okay, maybe sometimes you don't know what this particular protein is doing, but just because this protein is interacting with a repair protein, and it is a direct interacting partner, unlike maybe other proteins. Okay, then you may say that yes, this particular protein might also be doing a DNA repair like function. Okay, so that is what we can transfer the knowledge, and that is where the protein interactions can be taken up. And this dates back to the early chemical kinetics or lock and key model that we have learned during our high school. Enzyme lock and key model, I need not tell you, specificity and sensitivity is very important. So if there is a specificity that is seen in a molecule, it yields some kind of conservation. That's where it exactly fits. That bindability is represented in the form of the physicochemical properties. And of course, the systems biology based approaches need not always be interactions. It can always be associations, which means interactions per se. When I say interactions, it is not necessarily the physical interactions like this. Okay, which means if you have two proteins like this, it need not be physical like this. Okay, maybe a protein might be like this, where a domain of this particular protein might be similar to a domain of this particular protein, like this. Maybe a C-terminal here of this particular protein might share the C-terminal domain here from your window. Okay, maybe the domain might be the same. So then you may say that although they're not physically interacting, they share their domains, which means they're associated, which means they might perform similar functions. Okay, That also, you can probably take up. And this concept is what we call it as interactions and associations, which means when you say interactions are physical interactions, which means they're associated as well. But when you say associated, which means by advent of these particular things that I've shown as an example, say two domains that are shared between two proteins, that doesn't necessarily mean that, that doesn't necessarily mean that, they are physically interacting like this. Okay, they share the domains. Okay, which means they might have similar functions. Okay. So that now dates back to a simple adage: all enzymes are proteins, not all proteins are enzymes. Okay, something like that, isn't it? Likewise, all interactions are associations, but not all associations are not interactions. Okay, does it make sense, everyone? Yeah.
Yes, sir.
Any questions? Am I going fast or anything? Yeah. Okay. Yeah. I think my voice is now better, right? Yeah. Good. Yeah. So, yeah, let me see. We'll take the attendance at the end of the day. Okay. Oh, there are 26 participants, is it? Okay. Oh, good. Not bad. Yeah. Oh, gentleman sir is there. Thank you, sir. Yeah. So so likewise, based on the interactions, since we are saying that all interactions are associations, not all associations are interactions. There might be interaction specific methods as well. Sometimes not all interactions are similar, or they make up similar way they interact. So there could be homogeneous interactions or heterogeneous interactions. There might be organism specific, maybe in a fly, in a Drosophila, certain proteins might be interacting in one way. Their orthologs sitting in humans may not be interacting in a similar way. Okay, they might be interacting in a different way because they might have different domains. Okay, sometimes the proteins might be dependent on the core and the hypercore as well. So, for example, here, okay, this is my core protein, this MutSP, which is at the center. Okay. These are all other proteins that are at the vicinity. These are all the hypercore proteins. Okay, not all proteins are form the core, which means not all proteins are form the central here. Because this is central because it is interacting with almost all the proteins here, isn't it? Hence, it is a core protein. Whereas this is a hypercore protein because this hypercore protein is only interacting with this or maybe this, not with all others, isn't it? So that is where the difference between a core and a hypercore is. And you can also have different networks with organisms and different strains. And the networks sometimes vary based on the syntrophism as well. Syntrophic networks or food web networks, you pretty well know. A larger animal eating a smaller animal, and again a smaller animal eating a smaller and so smaller and other things. Okay. So, and there are different applications for the systems biology networks. Largely, we focus on the phenome interactions. Phenome is a phenotype. Okay, that phenotype might largely be interacting with an interaction, and that is where the functional relationships exist between disease-promoting genes, and all these kind of things play a very important role for various applications, including the drug interactions or protein-drug interactions, or even the precision medicine or predictive biology-based approaches. Then we can probably look at these kind of things. So we can say that the protein-protein interactions are quite common across all the cellular processes. You might expect a protein to be sharing the same domain with other particular protein. If they are sharing the same domains, then you may say that they are associated. Okay, but if they are physically interacting, then you may say that they are interacting. Okay, if they are physically coalesced, then you may say that those particular proteins are interacting with each other. Okay, and then of course for this to check whether or not we use different databases, and it all depends on the experimental type as well. And of course, you may definitely say that if two proteins are relatively closer, they're relatively homologous, then you say that they might interact in the same way. So which means you might expect a protein sitting in mice, say A interacting with say B, and its homologs, which are orthologs in humans, say A dash and B dash, they might also interact in a similar way. Okay, so that is where we might say that. This is my first network, protein interaction network, which we have developed in 2014. When we developed it, when there were no visualizers like Osprey or Cytoscape. But this was by my colleague Casper Lagay, who is now an eminent group leader. So what we have developed was, we developed this network based on a simplest interaction module. All square boxes are all our queried proteins, irrespective of the colors. Okay, like if this was a square box, this is a yellow one, is a mitochondrial one. But this was a yellow one, which is our query, but also in red color, which means it's a deceased one. Okay, as you could see, this is not ours. This is our circle one, but it's a mitochondrial one. Whereas here, as you could see, this is a bold one, which is largely pathogenic in nature, and it's a diseased one. It's also mitochondrial one. Okay, so we have such kind of networks, when we develop it. ENSG IDs are ensemble gene IDs. And then imagine this is only primary networks. Just imagine this is you have a Facebook network, right? You know what is a Facebook, right? In a Facebook network, you have, or in a LinkedIn or in a Facebook, you have one primary connection. Sometimes you will have friends of friends who want you to connect. You might have never seen them. Okay. You might empathize, you might sympathize and say that छोड़ दो यार, ये मेरा दोस्त है, दोस्त का दोस्त है, उनको भी connect कर लेते हैं, even without looking at the shekel, without looking at the face, right? So here also you might expect certain interactions, that are that are coalescing, where you might be having interacting partners of interactors, which you deem to find interactions. That is what we did, and this is a big slide, as you could see. So this is the second and third degree interaction networks, and during both cases there are no visualizers. So as you could see, I have denoted my proteins like this, searched it. What is that particular protein? Okay, manually. Okay, is an insulin receptor and other proteins which we have taken into consideration. And now you get family or friends of friends, those other interacting networks. And then we could come to a conclusion that this is our interesting node, which is node interacting with several other diseased proteins that are not mitochondrial in nature. Okay. So this is what systems biology, this is my first part of the talk. So systems biology, with systems biology has stemmed from the nascent bioinformatics. Protein-protein interactions play a very important role. Protein-protein interactions give rise to some functions of certain known unknown regions in the form of orphan genes as well. There are different tools and different layouts represent different things. Systems biology is not systematic biology. Systematic biology is a taxonomic biology. And you come up with a different union of interactions and you learn how these particular networks are known. Okay. And you can come up with different approaches. Okay. So I'll probably stop here, but I just want to give you a highlight saying that the bioinformatics is, as a bioinformaticist or as a native biologist, your main approach is to ensure that you lessen the scale of experimentation. Okay. You do some experiments, but as a bioinformaticist or as a systems biologist, can you probably come up with certain methodology where you can tweak your protocol and say that you can employ certain bioinformatics and come up with a conclusion, so that you need not do all those lumbar, experiments as a part of your approach. Okay. So I'll probably stop here, unless otherwise you have some questions, and I'll probably proceed with some hands-on. Yeah. Any questions, please? Yeah. I'll be happy to take some questions. Yeah.
Okay. Before somebody is emboldened to ask questions, I will ask a question, Prash.
Yeah, please, sir. Please, sir.
Yesterday only I was looking at that Phenome paper of yours with Sajita.
Yes, sir. Yes, sir.
And I was trying to get into the definition of phenome in Google. I couldn't get, but then I went to Google Scholar and found some papers. Right. Now, what my question is, in an interaction, phenome interactome, where does the phenome comes in the network? Yeah.
Yeah,
In the
phenome.
node or in it? Yeah. Right.
Yeah, I mean, yeah, that's a very good question, sir. Thank you for that. In fact, a phenome is a very, I would say, a very hackneyed term, because if we talk about a phenome, it's nothing but uh, an interactome of several phenotypes. But phenotypes are regulatorily caused by a protein. So what is that particular protein is represented in the form of that protein structures, actually. So we know that maybe some 10 proteins might be representing say diabetes mellitus. And now when we form a network for that, we may say that is a phenome representing diabetes mellitus. But we are not asking whether or not these 10 proteins alone are making up certain diabetes mellitus phenotype or phenome. We are asking whether or not within these 10 proteins, are there any other extra proteins that are known to induce changes in this particular phenome? So that is what we call it as phenome interactome networks.
But in my opinion, there must be some way of introducing the phenome information in the interactome, like the links to the nodes or
Yeah.
nodes themselves. Yeah.
Yeah, I mean, we do introduce them, but not at the data level. Of course, we can introduce that, but we only represent it in the form of a stereotype interaction network here. That's it. Yeah.
Right. Thank you. Yeah.
Thank you, sir. Yeah, that was my mentor, Jayaraman Baladi sir, who is a professor emeritus at Flame University, Indrashil University, and Savitribai Phule Pune University, Shiv Nadar University, and our past president of Bioclues and advisor in chief of Bioclues. Okay, and sir is 76-year-old. Can you imagine? Sir is very active and is going to take machine learning tomorrow, and I'm going to attend that. Thank you. Any questions, please? Yeah. So, assuming that there are no questions, I'll probably do some kind of exercises for you to do. We'll have a typical exercises here. Okay. So one is gene cards is a well-known tool. You can type say whatever gene of interest, say TP53. This is largely for the human genes. Okay. And you get some information. Alias is what is it called by other names like LFS1, okay, RP53, BMFS5, BCC7. These are all the aliases. There's something called human gene nomenclature classification ID, which is represented by this NCBI gene ID, NCBI gene ID. OMM is online Mendelian inheritance in man. A quick summary and all of those kind of things. You've got a lot of other things: antibodies, different sources, different functions, genomics, drugs, primers, clones, cell lines, tissues, pathways, animal models, mutations, variants, different things. But that is not what we want. So what you could do is, if you can select it, Control F and select interactions, okay? Because we are learning systems biology here, okay? So when you give interactions, you'll get some, small interaction study here. So it says that this is interacting with, TP53 is interacting with so-and-so genes and other things, okay? A quick summary, you can probably do this. This network you can also represent using, string, okay, string.ml.de, okay. So here you can search, this is the biggest network. You can search multiple proteins by names or even by sequences if you want. Okay. Say you can even search like this using NCBI, say protein, say TP53, say Homo sapiens, something like this. Okay. And is a Boolean expression. An operand, not Boolean. And you can use organism. Okay. Then you get some genes. You take RefSeq as a reference sequence, which is a reference sequence database. Okay, and you get some hits. And you get several sequences. So for example, if you want to take a FASTA sequence, you can even download, send to file, FASTA, and download. Okay. And so for example, if you want to take FASTA sequence, so how do you know that the protein sequence is a genuine one? It usually starts with methionine. Okay. Methionine is because it represents a start amino acid. So it starts with greater than symbol like this. Copy it. Okay. And then you go here. You can go for proteins by sequence as well. You can paste it here. If you want to have multiple sequences, you can also go for multiple sequences also you can upload it. Okay, and click search. You can also give auto detect here. Okay, so for example, you can give Homo sapiens if you want. Okay, but since I've given a protein name here TP53, I'll give TP53 and give, say, Homo sapiens and click search. Then it gives me that TP53 has got other I would say aliases. You see, other aliases are also known here. Okay. Which one you want to take? So I want to take the first one. Usually the first match is the best hit. Click continue. And it will generate a network here. So usually a network is usually in red font. Okay, this is your red font. So this is your network. Okay, and you see, as I told you, different edges or lines are there. One is purple, green, and light green. This is what it represents. So this is text mining, which means maybe TP53 and CREP might be represented in the form of a text somewhere in a paper somewhere. Light green is also there. Okay, it's also represented somewhere, and purple is also there. It indicates that it's experimentally determined, which means they might be physically interacting. Okay, so likewise, different networks, are represented. Okay, like this. Now this is only primary network, right? You have not taken the secondary network, which means TP53's CREP might be interacting with something else. That kind of network is not seen here. But before you get into this is how the functional partners are, all the interacting partners, and it is represented by a score, the total score. 0.99 is 99%, which means TP53 is known to interact with 99.9% score. And it's from experimental interactions, database interactions, and text mining interactions, which means they might be sharing the same database. They might be sharing the text mining. They might be physically interacting with experiments as well. Okay. So what we probably do here is you can also change the settings. Suppose you don't want to look into text mining interactions, or maybe you don't want to look into databases. You can untick it. Okay, and click update. Now you see, you have only got interactions that are, interactions that are only from purple and black. Okay, so black is co-expression, and the purple is the experimentally determined. Only the physical interactions have really come up. But for brevity, keep everything, because these are also very important. Okay. You can also change the settings. You can also change the confidence to 90%, 70%, 40%, or 15%. Or you can even give the custom value as well based on the number of scores that they are matching. But keep the default here and click update. So you got a network here, right? Now you click more. So when you click more, you are getting the second degree interactions. Now imagine in a Facebook, friends of friends, right? As I told you. So maybe known to be, maybe I'm a connection of Jiraman sir. Jiraman sir might be connecting with, say, Indrani. Indrani might be connecting with, say, Goldie. Maybe I'm not connected with Goldie. So where I am here, TP53, I can even get to know where is, that third degree connection as well. So this is what is all about when you click more. So you can keep on increasing that network, and you can make your network, quite bigger from time to time. You see, this much bigger network. Okay, and now we export the network and download it in the form of a PNG format. Okay, PNG is nothing but a network format. So when you download it, you can view the network. Okay, like this. So this is network. Don't worry about the block lengths. I'll show you how to remove that. Okay, you can download it, and you can also download this in the form of a table. Okay, like this, and then open this particular file in a Excel. Okay, like this. Okay, and this is what you get. So the first one is bold. Keep it bold. So you see, this is your node 1, which is your gene, and node 2 is the other gene. So your ATM is interacting with CD6, ATM is again interacting with SIT1 and all other genes. Node 1 is 9606 is a human protein code, human genome code. So it's interacting with another gene. So its node 2 is CD6 NSP ID. NSP is ensemble protein ID. Okay. Likewise, you are given this. And what you need to do is you take this bold and filter it. And now the combined score is there. Now you select this and sort descending. Okay, which means the biggest score, sort descending, not descending, sort descending. As you could see, the largest networks, ATM is interacting with TP53 with 99.9% combined score. Okay, but the experimental determination is 91.5. You see, this is an average. 91.5 plus 90 plus 98.9 by 3 is this much, something like that. Okay, this is how it is usually represented. Okay, so usually you have to trust only the experimentally determined interactions, because those are the genuine interactions, right? So if you have those genuine interactions, you can probably do that. Okay, those interactions can really be taken into consideration. Okay, so now you might want to ask a question. What is the cutoff that you want to keep? Usually 90% cutoff you can keep. So here you want to keep a cutoff. So here you want to take filter, standard filter. Okay, combined score less than equivalent to say 0.90, something like this. Okay, so or 0.99. I think I've taken others as well, right? So it is greater than I've taken, or less than equivalent to 0.99 you can take if you want. Okay, so that is the cutoff that you have taken. So you're getting all of them. So even you can even take another cutoff. Okay, you may say standard filter again, and combined score greater than equivalent to say 0.80. Okay, so you get only those interactions until 0.80. You see. So these you may say it as a bona fide interactions. Okay, so don't worry about this. I'm not saving this particular file for some reasons. I'll tell you later. So this is one type of network that you're going to construct. And then there are other tools like Gene Mania. Okay, but it works only on some select organisms, whereas in string you can use hundreds of organisms. Okay, this is only a human example that I've given you. So Gene Mania is a bit slow. You have to have a good Java in this. So please have some patience. So here you can use humans, Arabidopsis, Denio, fish, C. elegans, Drosophila, E. coli, mouse, ratus, and the yeast, budding yeast or something like this. So you can probably use this. Yeah, Gene a la carte doesn't work. It's only a paid version. Please, it's only a paid version, Yasin, unfortunately. Okay, yeah. So genecards.org works. Okay, Gene a la carte is for a different disease spectrum that you want to take. If you want to become a member, you can pay some money to them and you can do that. Okay. So here, let me take the same gene, TP53. Okay, you can use several genes if you want. Say I can use BRCA1, MLH1, MSH2, PMS2, whatever genes, all should be recognized. If you don't give some random name, suppose I want to give a gene called crash, okay, it doesn't recognize, you see. It says that it doesn't recognize. Crash is not a gene, okay. So it doesn't recognize. So if it gives click star, then you'll get a network here for humans, okay. And then you'll get a network. Yeah, you see this is a network, and you can click this circle. All the circle ones are all your queried ones, all the bigger ones. Okay, and these are all represented by different things. Usually the pink one is physical interactions, co-expression, predicted, co-localization, genetic interactions, pathway, and shared protein domains. Usually co-expression means two proteins are co-expressed, which means they are also co-localized, which means imagine in a cell you expect a nucleus containing some hundreds of proteins. They're usually interacting within each other in the nucleus, right? So which means they're co-localized, but they're also co-expressed. Who knows? They might also be upregulated in the same time. Okay.
Sir, what to do after selecting the gene?
Yeah, after selecting the gene, click search. That's it.
Okay.
Yeah. Okay. Where were we? Yeah. So what I was saying is, you have certain co-expressed proteins. They might also be co-localized as well, right? So then you have those physical interactions. Suppose you don't want certain genetic interactions or pathways or shared protein domains, you can remove it. Okay.
Sir, any way of selecting the organisms here?
Yeah. As I told you, this is how you have to do this. Yeah. I mean, this is how you need to choose the organisms. Unfortunately, there are only select organisms of interest here. Okay. So Arabidopsis is only there for plants, unfortunately. Yeah. So you can use physical interactions or co-expression or co-localization like this, and you get this particular network. Select, download network as an image. This name comes, outside. I mean, the gene name comes outside. That is an alternative image. Okay, but if it is shown like this, the name comes at the center of the node, something like this. Okay. Usually this is represented for your publications actually, okay? And then you can download the report. The report is usually in a PDF format, okay? And then once you download the report, you can see what are those interacting networks that you are, getting, okay? Yeah, this is how your, report looks, okay? And then these are all the different references and other things. Where were we? Sorry. Where were we? Yeah, here. Okay. So this is what you get from a report. Okay. But that is not it. Sometimes you want to see whether similar queried proteins might also be seen in, say, Arabidopsis. Okay. Which means, does Arabidopsis have these proteins where you want to transfer the network? When you click, let's assume that there are certain orthologous proteins interacting like this that might be present over there. When you click this,
I selected one gene from plant that is in Arabidopsis, but I'm getting network with all human genes also, I think.
Yeah, that is what I was trying to say, Yasin. You may not expect the orthologous proteins sitting across different organisms. That is what I was trying to tell you here. So, for example, here, I don't see TP53 here. You see, I've given the same thing. TP53 is not there. Okay, you see, TP53, of course, it's a tumor suppressor. It's nothing to do with Arabidopsis. I don't see that. But other proteins which are DNA repair proteins, MSH2, MLH1 are seen in plants. Okay. Interestingly, BRCA1 is also there. It's something
Yes,
related
sir.
to.
After seeing that only, I got confused.
Yeah. So this network exists. So what I'm trying to achieve here is something what we call it as, say for example, in humans, say you have a protein A that is say interacting with B. Okay. Suppose in Arabidopsis thaliana, you have an ortholog of A. So let's assume A dash. Likewise, you might be having an ortholog of B, which we because A is interacting with B, okay, then you may say that A dash is also interacting with B dash. So such orthologous interacting partners are called as intralogs. Okay, so this is what I was just trying to see if there are any intralogs sitting here. Okay, so obviously TP53 is not there, so it's not seen. Then you can download the report and see the network. Okay. There are several other tools which I'll tell you probably once I create the WhatsApp group. I'll tell you what exercises that you need to do. But we have a software called Cytoscape which I asked you to download. Okay, so here I use Linux, so I'm opening Cytoscape like this. Okay. It uses Java, so if you are using a Windows user, you can use a Cytoscape. Cytoscape is a visualizer, whereas Gene Cards, Gene Mania, String, these are all. Okay. So I have a what do you call? I have this particular visualizer tool like this. I can open it. I can import it. In fact, I've downloaded that table. Do you remember that Gene Cards table that I've downloaded? Where is that Gene Cards? Not Gene Cards, the string network, which I downloaded in the form of export. Okay, this one, export network as a table, which also opens in Cytoscape, where I have, what do you call, um, uh, check the combined score. Do you remember that thing? Okay, that I've downloaded. Now I want to open it. So file, import network from network from file. Okay, and then you download. Go to trash. I don't know where is the downloads. 1 minute. Home, trash, downloads. That file must be somewhere
Sir,
here. Yeah.
sorry to interrupt you. Sir, some participants are asking the name of the protein, sir.
Which protein name? Sorry.
Naveen, Naveen posted a question. Naveen, you can unmute kindly and you can ask.
I'll get back, Indrani. I'll get back. Please, I'll get back. Yeah.
Sure.
I'll get back. I'll get back. Yeah. Yeah. So this is what I'm opening this table, and I'm opening the network. And this is how my visualizer looks. Visualizer, the reason why we use visualizer here is there in Gene Mania or in String, you can only search, but you cannot visualize in different ways. So here I can go to layout, circular layout, something like this, you see. Okay. This is a wonderful circular layout I got. Okay. And I can find different views. I can put in grid layout, something like this. Or I can go for hierarchical layout. Okay. This is something like this. Okay. And I can also go for stacked node layout, something like this. But the best view is the circular layout. Okay. And I can select one node and ask what are those particular nodes that I've taken. And as you could see here, when you select this network, okay, you can get the node table here and also edge table, how many nodes are interacting and how many, and you can also get the network table as well. Okay. and you can download it. That's up to you. But this is not what we want. This is a visualizer. In a visualizer, you can keep this network or this node like this, 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 whatever. And again, if you want layout, circular layout. Okay, all nodes. Okay, this is what you wanted it. This is why you use a visualizer. But in a visualizer like this, the most important challenge is now you want to ask a question: which node is a hub node? Hub in the sense, which node is making a very important commodity in a network, or which node is influencing the other nodes. Okay, that is what you wanted to see. So for this, you have to go to apps, app manager, install something called CytoHubba. Okay, there are different apps, but CytoHubba is a very important tool where it measures the ranks on which node is important or which protein is important. Okay, so CytoHubba is a very important thing which I already installed it, so it's already there as you could see here. So I can go here, and this is a table which is already. This is always known to be interacting with each other. Are these two nodes interacting or not? That is betweenness. Closeness is if this is a network and if this is a node, is this particular node always interacting with this when this is there? Okay, that is what we define by the closeness network. Okay. And there's something called clustering coefficient. Clustering coefficient is a simplest statistical method. How many clusters in a simplest regression based method, how many clusters that these particular proteins can really make every every aspect of it. That is denoted by clustering coefficient. So by default, we'll go for the clustering coefficient and we'll find the top 10 ranked genes. You can even make it top 20 as well, depending on your choice. But by default, I want to give top 10 and choose clustering coefficient and click submit. Okay. Now you see, I can click layout and again circular layout. And now in a circular layout, the brightest color or the brightest node color indicates better interaction network or better ranking. So as you could see here, CSTF1 is rank number 1. It is a loner node, but it is not known to be interacting with others using clustering coefficient. But others clock is known to be there, but lesser the color intensity, lesser is the rank. So SMAT2 has got the lowest rank here. Okay, whereas CCNA1 is the next least rank. So the more body, better the rank it is using the clustering coefficient. Likewise, you can also choose a betweenness centrality, as I told you what is betweenness, and click submit. So you see the beauty with betweenness here is when you chose layouts and click circular layout, the top rank is TP53, as you can see here. Okay, TP53 is the brightest, which means it is making a biggest hub. This is what we wanted. So always, betweenness centrality plays a very important role. And as you could see, the other ranks are always denoted with different color nodes. Okay. Likewise for closeness, click submit. And here again in closeness also TP53 is top ranked. Okay. So this is how the nodes can be represented. You can even increase it to 20 nodes if you want with the same thing and click submit. And again you see if you click with 20 nodes, your network keeps on increasing. And you can save this network as a file as TP53. Sys file is cytoscape file. Okay. And you can even export the network, export network to image. Okay. You can increase it to bigger pixels in a PNG file or JPEG or whatever you want. Okay. And save it. And you can use it for your image and you can write a report. Okay. So my take-home message here is we have used betweenness, closeness, and clustering coefficient. Clustering coefficient is the simplest statistical approach. Betweenness is what's the likely chance that two nodes sitting next to next might be interacting. Closeness is maybe several nodes interacting with each other, but what is the per chance that if its adjacent node is also interacting in a similar way is represented by closeness.
Sir, what is the advantage of using the cytoscape than the string? Directly, why don't we use string results?
Yeah, that's what. String doesn't allow you to rank the nodes like this. It doesn't allow you to visualize in different colors. So cytoscape is a visualizer, whereas gene cards or gene mania or string is a normal database tool. If you want to rank it, if you want to make a different visualizing like this, like I want to make a visualizer of, say, in the form of a hierarchical network like this, I can probably use cytoscape.
What does that mean actually?
It's a visualizer. It's a visualizer, Yasin.
Sir,
So
that is clear, sir. That is clear. In this picture, how to interpret, like what is in
right.
the real or
The interpretation is based on the ranking. As you see from the cytop, I've known that TP53 is rank 1. So my TP53 is here. It forms a central hub. You see, when I click this, it is known to interact with almost all the nodes. Okay? Which means that is the reason why it is a hub gene. It is a top rank gene. Okay. So whereas CDK2 or ATM, okay, it's not probably interacting with so many genes as others. Okay, that is the reason why it is ranked 4. Or whereas my other gene like CCNA1, okay, it is only interacting with 4 genes, as you could see. Okay, so it is ranked 20th. So it is not making that hub when compared to the other proteins.
Okay.
So I thought you will probably do this exercise. Any quick questions? Please protein degree parameters Pragati. I would definitely do these exercises and finish these things. I hope you're all live members. So this is only for live members. I would now open this this particular thing. All of you, please. What you do is, I'll keep the attendance. So you write, all of you, please write your name. July 17th, 2023. Say, for example, Goldie. Okay, I'm giving her score as 1, which means she has attended this. Okay. Likewise, we'll keep on adding the attendance. So please write your names here, all of you. Okay. And we will double check whether or not you're a live member, and then we'll send you the link for tomorrow. Tomorrow we'll have Jiraman Valadi sir's talk. Okay. So thank you very much. There are no questions.
This file is already sent to us.
This is shared. This is shared with you all. Let me share it again. Yeah. This is here. I've shared with you all. And let me create a WhatsApp group as well. I don't know if there is a WhatsApp group that I've created earlier. I don't know. I don't see some of my groups here.
So we will write our names, now phone numbers also, WhatsApp number also here, so that we can.
No need. I'll give you the link for this. Please wait. I shared the link. So all of you, please join this WhatsApp group.
Hello.
Thank you.
Hello, everyone. Good evening.
Yeah, good evening, everyone. So let me please allow me to introduce other speakers as well. So we have Pushpendra Singh, who is a principal scientist E2 at Tribal North NIRTH Institute, based in Jabalpur, Madhya Pradesh. He is our research chair. We have Anil Kumar, who is our outreach chair, who is a professor at Vignan University. We have Sri Manjari, who is a secretary. She is a senior scientist in Power DX in Hyderabad. We have Indrani Biswas, who is our joint secretary. She is ICMR senior research fellow in Pondicherry. We have who else to introduce? That's it. I have already introduced Jayaraman sir. Yeah. So these are all the other faculty, and we'll probably see you tomorrow. Yeah. Thank you so much, everyone. So please do those exercises, and I'll share you the links, for all these tools. Okay. Please feel free to ask questions in WhatsApp group, and please write your attendance so that we'll fill up the attendance. Thank you so much. Sorry, Manjari and Anil, are we meeting here for the meeting or in the another link?
The same meeting link, I guess.
Okay. Yeah. All others, please leave. We have some core meeting, please. All others, please leave. Thank you so much.